So tonight, something a little bit different. Amy presenting Unlocking Past Environment and Ecology with Biomarkers from Microbial Communities. Exceptional preserved fossils from the 50 million-year-old Green River Lagerstatte. Cool, no worries, thank you.
Hopefully that means a bit more to you, that title, by the end of this. But the research here has been done at Curitn University in the School of Earth and Planetary Sciences, but particularly in the research group I'm a part of is the Western Australian Organic and Isotope Geochemistry Centre. This is part of a wider project, a wider fossilisation project being organised by my principal investigator, Professor Curitn Bryce. Some of you might know her. But thank you for having me today. I understand there is organic geochemistry in this, and I have to apologise for that, but there is also fossils. So hopefully you'll be kind of kept entertained through it. So I'd just like to acknowledge the traditional custodians of the land we're meeting on, the Whadjuk Noongar people, and pay my respects to elders past, present and emerging. I'd like to recognise the strength, resilience and capacity of the Noongar people where Curitn University is situated. For me, I've always found it really impressive that the Indigenous people here, they still told stories of being able to walk over to Rottnest, and that's sort of representing that sea level rise and fall that we see globally and the last glacial maximum. And they actually still pass that on, which I just find really cool. Well before at least Western scientists identified it in Australia. So, for those that don't really know me too well, I'm currently a research associate with the Organic and Isotope Geochemistry Centre at Curitn, and I'm working on the ARC laureate-funded fossilisation project. I've been doing that for 2 years now, but before that I did my university at the PhD of Southampton in the UK at the National Oceanography Centre, focusing in on past climates, geochemistry, particularly of continental paleoclimate of the early Cenozoic. And before that, I did a master's in geosciences, focusing on aeolian and fluvial interactions, mostly for reservoir characterization. But basically, all of my work is rooted in geological fieldwork. Sedimentology and geological fieldwork, I like to kind of bring that line through it, but I think most of us like to get out to the field. This is Monument Valley in Utah. The rocks there are about 5 times older than the rocks I'm going to be talking about today. But I like the analogy of a road map through the presentation. So this is why this picture's in here. So I'll go through the introduction of the project and the reasoning behind it. Also how we gathered the samples, prepared them, the methods used for those unfamiliar, and the results and the importance, kind of where we're going as a group with this research, and then I'll hone in on my conclusions. So, Green River Formation. For those of you unfamiliar, it's a set of ancient lakes spanning Wyoming, Utah, and Colorado in the USA. It's approximately, the lake system lasted for about 10 million years when you consider the first occurrence and the last occurrence. And it actually hosts the world's largest oil shale. It is thermally immature, hasn't been heated enough, but it does host the oil shale in the southern basins, and a lot of evaporitic deposits in the northernmost basin. So the greater Green River Basin, that's a lot of evaporites. And in the south, you get these oil shales developing that are really quite famous. But in this tiny little basin up there, the fossil basin, that's where you get the majority of the exceptional fossils of the Green River Formation in the smallest basin. The nice thing about these lakes, they're not particularly badly deformed in any way, and they're mostly carbonate mudstones. A lot of the lakes are quite alkaline, actually, through their history, and they didn't have a lot of burial underground. So the basins were mostly formed through intercratonic sag from the formation of the Rocky Mountains and the Colorado Plateau, but also the uplift of those mountains would have had associated lithospheric flexure. So the weight of the Uinta Mountains and the Rocky Mountains would have forced basin formation as well. So I guess the main point of this slide is that there's different sedimentary expressions through the different basins. And even though there are hundreds of organic geochemistry studies on particularly the southern basins because of that oil shale potential, there's not a single organic geochemistry study on the fossil basin and why these fossils are so well preserved here or what was happening in the environment or the ecology. So that kind of sets up, I can't believe no one's ever actually looked at the fossils for the geochemistry. And for those that haven't worked much in the Cenozoic, and that's pretty young for us geologists, the early Eocene was actually the hottest point of recent geological history. Recent being after the dinosaurs died, or the non-avian dinosaurs, I should specify. So the Green River Formation actually spans what this hot interval that we call the early Eocene climatic optimum. We can tell that both through correlation of negative carbon isotope excursions in the Green River Formation, particularly in, these are found within the southern basin, not in the northern, but we've managed to correlate negative carbon isotope excursions to the marine realm, but also we have ash layers interspersed through the different basins that have been dated. So that's great. However, these were actually, in the southern basin, were flash flood large sandstone deposits. So we know that during the hottest part of the climate, at least in those southern basins where you get the oil shale development afterwards, you get massive, massive sandstone bodies during a time when the world was super hot and super wet. There was a lot of rainfall, essentially. Because it's so well dated, we do like to use the Green River Formation as potential, the early Eocene as potential indicator of future warming. And if we can understand the paleoclimate back then particularly well, we may be able to understand future warming a bit better. Yeah, and also I guess a point to make is that there's no permanent ice at the poles. And also, I'll just mention this middle figure. For those that aren't all that familiar, down here is, between the green and the purple, is the death of those non-avian dinosaurs. Up at the top is the modern day. And that's a stack of benthic, foraminifera, and oxygen isotopes. That proxy is used both for temperature, but also for ice loss and ice amount. So in the hottest part of recent geological history, the isotopes are actually very depleted. So we know that it was probably a fair bit warmer, just for people that haven't maybe looked at oxygen isotopes before. So, the nice thing about the Eocene, particularly the early Eocene, is it's not so different to what we understand today. The world kind of looked like this, or probably did sort of look like this. Big differences being that Australia hasn't fully unzipped from Antarctica yet, and South America hasn't separated from Antarctica either. So the Antarctic circumpolar current probably didn't exist as it does today. And India is still colliding with Asia. So you don't have that massive Himalayan front that would change the air patterns too. So those would be the sort of current differences as such, water and air current differences, there would be some. But aside from that, it's actually, you can still recognize a lot of the continents, right? You can still see Africa, you can still see Southern America, and sorry, and you can still make out North America. So that's quite nice for us to remove some of the perhaps geographic uncertainties when you go really deep in time. So we'll move on to talk a bit more about the actual fossil basin of the fossil rich layers. It's the smallest basin in the Green River Formation, but does host the vast majority of the exceptional fossils. Out of the three members that are present in the fossil basin, the fossil butte member is the most fossiliferous. What you're going to learn here is that Americans aren't great at naming things. Apologies if there are any in the room, but it's going to get a bit repetitive. So you have the fossil butte member in the fossil basin of the Green River Formation. So during the fossil butte member, it was the deepest and it was stratified, we're pretty sure of that. And there was continual sedimentation in this basin for about 6 million years, aside from perhaps small local erosional events like turbidites and things like that. But pretty much there was no exposure and erosion down, you know? Oh, wait, and I forget to mention some of the common fossils. Oh, well, some of these aren't that common, they're very rare. You have, they reckon, over 1 billion fish lived in the lake during its existence. They have some fantastic crocodiles, turtles, and some of the earliest examples of freshwater stingrays present. You can see how detailed and fine that's been preserved. It's also, as I mentioned, the shortest lived lake of the Green River Formation. And these are some of the actual terrestrial fossils that we find preserved in there. You have a really unfortunate early example of a horse. They're about dogs, like small dog size back then, but that obviously drowned in the lake. You get loads of plants. And the earliest example, I think, of a bat they found so far. And massive, massive snakes, because it was a lot warmer, so they could get really big. The nice thing about the palm fossils actually is that when you freeze in the winter, palm trees don't tend to grow and don't tend to reproduce too well. So even though you see palm trees like I don't know if you've seen them, but I've seen palm trees that grow in England, but it doesn't mean that they're thriving or that they're happy. They probably won't reproduce. So the fact that we've got loads of palm fossils and big ones too tells us that it couldn't have been freezing during this time period in winter, because they wouldn't have been successful, essentially, compared with all the other flora. Also, the thickness of the annual layers have been estimated from tufts through it, and they reckon it's about 0.2 millimeters per year. So it's quite decent per year. And all of these amazing fossils have been used to try and understand North American land mammal evolution after all of the non-avian dinosaurs were killed. And we had those niches for them to expand into, and luckily lead to us being here today. So it's been pretty helpful, at least for the paleontologists. And as far as age dating, I'm sure some of you know much more about getting ages out of rocks than me, but they used Smith et al. He's got this whole group, and they just spend their whole careers dating everything that they can in North America. It was done through potassium argon dating of biotite and feldspar crystals that were found in volcanic ash tuff layers. And that actually dates it to about 51.97 million years, give or take about 0.02 million years. Which is, and the geochemistry of those tuffs that they find in this basin and in the other Green River basins, that's been traced back to volcanic centers north of the Green River. So you're getting up into north Wyoming and current Idaho, lower Montana. That was really volcanically active in the early Eocene, which is great for us. So a lot of them came out. So at the base of the Green River in Fossil Basin, you had the Road Hollow member, which was pretty fresh water, but not particularly expansive. And then it became deeper in the Fossil Butte member, and it was at its lateral maximum, so to speak. And then in the Angelo member, it slowly got more and more saline and kind of dried out, became a puddle, and died. It's surrounded by the Wasatch Formation, which is essentially the kind of on-land contemporaneous equivalent of the Green River Formation. It's mostly alluvial fans and fluvial systems. So that's the on-land counterpart. And if you go to different basins, because the Green River is spread between 3 states, each geological survey is given the same formation, a different name, because that's logical. But it's essentially genetically, the Wasatch Formation can be found all around the Green River basins. They'll just have a different name probably, but it's the same environment. And based off all the wonderful fossils that have been pulled out, you get all these beautiful reconstructions that look like this, purely based off the macro fossils. There's actually never been any geochemistry or organic geochemistry done on this basin. So we don't know about how the water column looked, the geochemistry of it, how much oxygen there was. All these things are just based off the fossil discoveries. And there are a lot, but it's just very surprising to us, at least, that no one's done the basic organic geochemistry just to check it out, considering you have hundreds, maybe thousands on other Green River basins that have a lot of money potential in the oil shales. With this in mind, I set out to the Western US to do field work. I actually stopped by the Field Museum in Chicago first, which is on the right. Lance Ground kind of has found loads of species in the Green River. He might be the foremost paleontologist of this formation, and he finds things like this. And now I was inspired, and I went over to the Western US, but I didn't think I would be able to pull back anything quite like this. And if I did, they definitely wouldn't let me crush it up to put through a mass spectrometer. But despite that, I landed in Salt Lake City and then tried to go around the different basins to get fossils from all of them. So fossil basin has all the exceptional ones, but there are still some fossils in the others. So I also wanted to try and get them back for comparison. And we did get soft tissue preserved fossils like this, hopped out on the suitcase on the way back. They're all wrapped in foil to stop from contamination. But this hopped out and you can see the soft tissue and then the spinal column all the way out. I also collected different insects, found some plants, a bit of turtle bone over here. It's actually from a turtle plastron, like the base shell. And 4 different species of fish and a bunch of coprolites as well. So it was actually a really good trip. I felt quite lucky I was able to find all that stuff. But the focus of this talk will be specifically on the layers from the fossil basin, so the best basin. I'm focusing in on essentially the sedimentary layers in the fossil basin and trying to understand the geochemistry specifically of that. So these layers obviously were buried, but then we've had local erosion, the Colorado plateaus uplifting and eroding, so you've got these fossils now exposed nicely at the surface. The important thing about fossil basin, I guess for geologists, is that it's a terminal basin. So there's no outflow of rivers. Any water loss is through evaporation. So that concentrates things in there over time. There will be fluvial input, but during deposition of the fossil layers, it was all basically limestones and carbonate mudstones. Whereas in the southern basin, it's down here, at the same time you're getting these massive sandstone deposits. So we understand there's probably a lot of rain during this time. It's hot and it's wet. It's a hot, wet climate. But in the south, the rivers are actually provenance found that they're coming all the way from Arizona. So massive flash flood systems. And then there's a big bunch of mountains here. So we're just getting runoff from the local mountains. It's different to the other Green River basins already in sedimentology, but we just get carbonate mudstones. So we have organic rich laminated micrite, and I went and sampled at Smith Hollow Quarry, which is about 5 kilometres south of Fossil Butte National Monument, because it's quite nice. They've actually protected a bunch of land there with all the fossils in it. And this is I went to a private quarry just south of it, where they're very happy for scientists to come along and sample for free. And yeah, so that was very lucky. The quarry itself, as I mentioned, light beige mudstone, and you find dark fossils in the plains of these. Specifically, this talk is about the 18-inch layer, one of the most famous layers of the fossil beds. And there's a very low energy lake sedimentation, parallel laminated. In the top picture here, you can see lots of scree falling down from how soft those rocks are, but you can see the parallel beds. And the best time to hunt for fossils is actually towards dawn and dusk, because the light shines and you can see the spines, the backbones of creatures really well. So you can sort of see it here. But at dawn and dusk, with the lights low, it'll actually show up really obviously. At this point, you might be asking, why bother? Why go out and get sunburn when you get the Colorado Plateau? So you're already a kilometre up and you forget it every time. Aside from any excuse to get out to the field, because I love that, why not just take museum collection samples, right? Fieldwork, 1, but 2, a valid reason to non-geologists would be a lot of museum collections the pieces can be fairly contaminated. If you've ever been out in the field with paleontologists and they pull the rock out, they almost immediately will put some fixing agent on it, some kind of glue or adhesive that keeps the fossil all intact, without crumbling, without breaking. And that obviously is full of organics because it's a type of glue and adhesive. And that would mask any signal you're trying to get from the original rock. Also, in museums, they can be handled quite a lot, and you have oils on your fingers. Sometimes they're wrapped in waxy paper, and other times fossils could be used for outreach, so you don't even know if a kid's licked it or something. So there's something nice about going to the field and getting your own fossils, and you know exactly what's happened to them. If there is contamination, maybe it's something, but you know it's not had this 40-year history of who knows what. Sometimes people write in crayons on them because of the wax, and it's non-destructive, but wax is really organic. So yeah, that was one of the main reasons. The first reason is obviously because it's field work, so everyone loves that, right? So the fossils specifically at this talk are, I'm looking at the sediments of this fish and of this fish. This is just a fish face, but they're both the same species. So that's kind of nice as an equalizer, right? We're looking at the sediments from beds that have the same species in it. In the top picture there, that thin one, you can actually see the circular. That is cut through a spine of a fossil, and you can still see how circular it is after 52 million years. It's not been much deformation with these rocks, which is fantastic. So yeah, the focus is to understand the sediments around it so that we can characterize the paleo-environment, the ecosystem. Obviously, we're working on the fossils too, but when there hasn't even been a paper out talking about the general organic geochemistry of the basins, then you don't want to hop into the fossils without understanding the sediments in them. So, and before we do the organic geochemistry, you probably want to know what the minerals are as well. So I'm sure many of you are familiar with XRD to understand physical properties and phase composition. But we found, surprise, surprise, that the major rock forming mineral is calcium carbonate in a mudstone, in a carbonate mudstone. Can you believe it? And a lot of that would be forming through precipitation. So it's as you get evaporation of the water, you get concentration of calcite and calcium ions, and you supersaturate the pore space in the water, and you get precipitation of calcium carbonate. We also got minor amounts of anchorite and quartz and dolomite, and a lot of these would be probably eroded from the highlands. It's quite nice. We have granitic highlands, which align to the anchorite. And that was, they'll be eroded out of those highlands and then transported in through fluvial systems. So we have quite a low amount of quartz. And this got me thinking a little bit, if the quartz is higher in some layers, perhaps that could be a proxy for calcium carbonate precipitation rates. You know, so lower amounts of quartz means higher amounts of, or it could also be linked to seasonality in weather. So, you know, wetter summers, bigger rivers, more quartz. So there are variations, but it was nice to see it wasn't just calcium carbonate. There was actually a little bit of other stuff, but not much. We also did some program pyrolysis, essentially rock eval, because it is a sedimentary rock, and this is something that the oil and gas industry tend to do as standard anyway. And it's done on the kerogen, and kerogen is the insoluble, the non-dissolvable organics in a rock. They won't come out very easily. You essentially have to, this analysis gives you the petroleum potential and the thermal maturity of a rock sample. And it's progressively heated under inert gas, and hydrocarbons are released and carried away and analyzed. And what we got here is that we got something called a very low hydrogen index, and that measures the hydrogen to carbon in molecules. A very high hydrogen index and a very low oxygen index. And this is a Van Crevelen diagram, and that actually means that we plot directly up in a type 1 kerogen. These are more your sort of coals and your marine oils, and then the top type 1 is quite a typical lacustrine shale. And that's nice to see. It's very common in lake environments. It also usually means that it's got a lot of algal productivity, and the algae is contributing to the organic matter. And quite nicely, we also get an total organic carbon measurement of above 2, which means that in the oil and gas world, this would be a great source rock if it had been buried deep enough. So actually, it's pretty organic rich. So, organic geochemistry. How do we actually get the samples, and how do we extract it? First, I crush up the rocks, and then I essentially extract it over 72 hours using a mixture of dichloromethane and methanol, and those organic solvents actually extract the organic matter. So for over here, what happens is you heat up the organic solvents, they evaporate, they go up. These white things are called thimbles, and they're made of glass wool. And you put your sample in there, you weigh it. As this evaporates, it moves up, it saturates the thimble, and dissolves out the organic matter, it keeps on going up, condenses, and then gets transported back down into these. So over 72 hours, you're concentrating the organic matter in the round bottoms at the base. You can see a bit of colour in this one. And then actually, in these ones, you can see more colour. So that's actually the organic matter providing that colour there, that brownishness. And organic geochemistry is still a relatively new field of science compared to the others, and it's very much has been driven by the oil and gas industry. So, yeah, and it's also always important to have a blank, just because what if you're introducing organics to your samples? There's always blanks being run alongside all of these. And then once you separate them, you should be able to run a range of geochemical techniques on them. So just this is important for later in the talk. The organics in these little bottles, after I've evaporated down this mixture, this is bitumen, it's the extractable organic matter. But left in the thimbles in that sediment residue will be the kerogen, which is unextractable. So there's two different types. Now, we separate the compounds using column chromatography, and that essentially separates your total organic matter into lipids and pigments from least to most polar. This is because you can't run everything on the GCMS, a gas chromatography mass spectrometer, and you need to do cleaning stages before you put these things on your instruments, your very expensive instruments, I might add. So we use organic solvents of increasing polarities to separate them out. So your saturate fraction would be your least polar, and then obviously your polars will be has the most things on them that could get stuck in the column essentially. After this, we analyze it on a gas chromatography mass spectrometer, but we also have a much more sensitive one. That's a triple quadrupole. That means we can analyze even further. This will make sense when I show you the data, but I'm just reiterating it again. And of course, you would, with your columns, you'd run a blank one as well to see if anything's being added at that stage by your analysis. So, biomarkers, for people that aren't that familiar. They're essentially molecular fossils of lipids and pigments, and you can directly relate them to a precursor molecule. Usually it means that some biomarkers can be diagnostic for specific groups, or a specific organism, or a specific environmental signal. And in sedimentary environments, the lipids that do escape remineralization and be broken down, they're commonly reduced to relatively basic hydrocarbon skeletons. And these skeletons can remain intact for over hundreds and hundreds of millions of years. I think the best example, at least in organic geochemistry, would be this cholesterol biomolecule, so a modern molecule. Through diagenesis, it would lose the alcohol group, but also this, which is a double bond. So through diagenesis, you lose your alcohol groups, your ketones, and any unsaturated molecules would become saturated. Essentially, you're simplifying it down, and things are breaking off. So that's after that's happened, you then get the biomarker cholesterol. So that's probably what we would recognise as organic geochemists, but it means that the previous thing was cholesterol. It's kind of like an inheritance in a way. I'm sure you're familiar with the tree of life. This is like a biomarker tree of life. Today, we're going to be focusing on the bacterial branch with biomarkers called hopanes that are bacterial in origin, but also eukaryotes for sterones, which represent animals, some plants, and fungi, and also biomarkers, as well as telling you what's living in the lake, they can also tell you a little bit about the environment, depending on what biomarkers you have present. So, ready for data? You can say no. So I'll take a sip and allow you to feast your eyes. So, this is talking about the saturate fraction, that simplest fraction, the first one that you extract out. And it's very nice because what I just showed you, that domain of life, you can see most of them here, actually in this trace. Put some pictures help, but you have bacteria, so the hopanes, you have bacteria. This big peak here, the C31 outer beta 22R hopane, that's specifically linked to cyanobacteria. So you can even go into the families and find different ones present. We also, over here, this you get pristane and phytane preserved, and they are from the side chain of chlorophyll. So that we know they're from photosynthetic organisms living in the water column. And they get preserved differently depending on whether you have oxic or reducing conditions. So, pristane, which is tiny here, that's favourably preserved in oxidative conditions, more oxygen-rich conditions. Phytane, which is very high, is present in reducing conditions, low oxygen conditions. So we can already tell from this that you probably have low oxygen in the bottom water of the lake. If you move up a bit, you have these what we call mid-chain alkanes, which are actually from macrophytes, so like sea plants that are rooted. So we have that present in the lake, which is nice, and that was probably washed in. The biggest peak is from this one here, and that is actually from leaf waxes from land. So leaf waxes that grow on leaves, they protect the leaf from the chlorophyll, from UV, from disease, and they're very resistant to being broken down and decayed. So they've actually, as the plant shed some leaves, it's gotten into the fluvial catchment, and it's been washed into the lake. Maybe the rest of the leaves aren't, but the leaf waxes are really robust. So we have such a small elliptical lake that it's not surprising to me that we're actually concentrating loads of plant matter in there. But it's the biggest one, which I was a little bit surprised at, being honest. We also have, as you can see here, from the 23, and 24, 25, and 26, you can see that the odd ones are far more predominant than the even-numbered alkanes. And again, that tells us that we have a lot of land material being washed into our lake. So just in this one saturate fraction chromatogram, we can see that there's a lot of mixed organic material present or being preserved in this lake. So we get all these different ones. But for some reason, there's a lot of land plants. Now, stearains, still in the saturate fraction, stearains are to do with the eukaryotes that I was talking about. Animals, certain types of algae, us, not us back then, but essentially, if you look at the data, this base one here is just run on a regular gas chromatography mass spectrometer. But the ones above that have colours, that was actually run on a multiple reaction monitor mass spectrometer, which is much more sensitive. So it can actually separate out different ions. So at the base here, you can see there's a lot of overlap between the compounds. There's a lot of coelution. So essentially they have the same mass and the same charge, even though different things produce them and they look different. So they'll overlap on this 1D line. An MRM, as we call it, it uses the parent and daughter transition ions. So you can actually separate your stearains into C27 at the start, C27 at the top, C28 in the middle, and C29 just here. And they all have different sources. So C27 stearains are predominantly from animals and red algae. Your C28s, which are a smaller constituent here, your C28s, they're from fungi and several groups of algae, including diatoms and coccolithophores. And your C29 stearains down at the base here, it's meant to be green, that's actually from higher plants, typically, and chrysophytes, so golden algae, if you've heard that, and also green algae. So you can actually separate out different areas of life. But what we see here is that, again, the higher plants and the green algae is dominant, although not that far behind are the animals and the red algae. There's a lot going on in this lake. We also, quite interestingly, see these tiny little bumps up here? These are called diasterones, and they're incredibly low in these samples, which is nice because that tells us probably haven't had that much heat and pressure, thermal maturity. There's also, you can see the numbers and the letters here. We have a lot of, the biggest peaks are from the alpha 25 configuration, which is the sort of least mature version you can have. The R configuration of a molecule, that is biologically produced. Over geological time, that will turn and become an S configuration. So that's geologically favoured. The fact that we have so little S isomery present basically means that it's not been buried under much heat and pressure, which is great because that means that our organic molecules probably haven't been broken apart and messed around with loads. So that's good news for us, actually. Just to move on to the hopanes, there aren't so many slides like this, I promise. Just to move on to the hopanes, instead of breaking at 217, as you saw the numbers along here, it actually breaks at 191 preferentially. And again, we had coevolution in the GCMS data, but when we put it on that more specific MRM, we could separate it out into C27, C29, C30, and C31 hopanes. And what's really nice here again is that we see the C30 and 31 dominance, which means that we have a massive amount of bacterial presence in the lake being preserved. And we also, in the top one, if you look at C27 hopane, similar to those diastereones I was talking about, you have TS and TM, which are two biomarkers. I guess the name for TS is technically trinorhopane, and then TM is trinorneohopane. So they're the same molecule that's been changed. And essentially, yeah, the total lack of TS means that it's really thermally immature. So again, great. We've got things from different areas of the tree of life, bacteria, animals, plants, all agreeing that thermally there's not been much happening and all the molecules should be well preserved. So that was really nice. I guess the important little thing that we notice is the presence of this molecule called hemastobrain. It's a bacterial hopane that's produced from bacterivorous ciliates, so creatures that would live at the chemocline and prey on bacteria. They would take in tetrahymenol to form sterols, and essentially it's that molecule that's preserved over time. And it's a very long way of saying the presence of this molecule tells us there had to be some chemocline, therefore salinity stratification in your lake. So that's really nice. It's like, okay, there was salinity stratification of the water column. Great, that wasn't known before. This lake, everyone calls it freshwater, but there was actually, in a layered sense, there was saltier layers. This is just pretty rough, quick, new data. But pyrolysis, that's when I was talking about the bitumen and the kerogen, and I made that point. Essentially, in pyrolysis, you take the powder that's had all the organics extracted out of it, and there's organics still locked in it called kerogen. So we have to use very high temperatures under no oxygen. Essentially, it's like a little controlled explosion to force that geological process of heat and time. And basically, it cracks out the other organics. And what's nice from this, actually, is that we see a different array of N-alkane and alkenes, those unsaturated molecules. They're dominated by the C8 moiety, but they go all the way down to the C34. And the interesting thing here is that at least this profile, this matches with other Green River basins. None of the other geochemistry has so far, but the kerogen, the locked up stuff, so it's actually pretty similar to previous work. So that made me not that interested in it. This is actually some previous work by the group I'm going to touch on as an introduction before talking more about sulfur stuff. Yeah, previous work in the group, and it's about aromatic carotenoids. So we're going to move on to that middle fraction, the aromatics. And aromatic carotenoids are tremendously important biomarkers because the only large biological, significant biological source we've found so far are phototrophic green and purple sulfur bacteria. So it tells us that if we have the presence of these in sedimentary environments, because most phototrophic sulfur bacteria, they need light and they need reduced sulfur as electron donors, but they also need the absence of oxygen. So it tells us you had to have if there's the presence of these biomarkers, you had to have a water column where it was sulfidic, but also no oxygen, but within the photic zone. So that's not particularly nice either. But we also, yeah, so we know it's salty, and if you get these biomarkers, then you'll know that it's sulfidic as well. They're really common in microbial mats as well. So if you've ever been up to Shark Bay and seen those mats, they would have been stuffed full of sulfur bacteria. Yeah. With that heavy-handed introduction, guess what we found? We actually, this is the aromatic fraction. These things are called MTTCs. That's a spoiler. We'll get to that in a few slides. But importantly, we actually found isorenitane in the aromatic fraction. And yeah, the predominant source of this is sulfur bacteria. So we were like, okay, we've got the presence of this, but we really need to try and find the rest of them if we're going to be definitive about this and say there was photic zone euxinia happening in the fossil basin. What's interesting in this aromatic fraction is we found no beta-carotene. And that's a biomarker that tells you at massive algal blooms. It was also first discovered in the Green River Formation and has been found in every organic geochemistry study of the Green River basically since this one. So this one hasn't found any beta-carotene, no large algal blooms, which to me also makes a bit of sense. People were kind of shocked I didn't find it, but I'm like, it's a really productive environment full of fossils like turtles and crocodiles. If there were frequent thick algal blooms, that would have totally destroyed the ecosystem of the lake. So I was like, I think it makes sense that we don't find it. We also found a biomarker called perylin, which is a product of quinone pigments from basically wood, breaking down wood, fungi that breaks down wood. So that must have been coming in from the terrestrial realm. So we know there's a lot of microbial activity, but also terrestrial input. But just to finish off with this, we have potential photic zone euxinia. So we had to follow that up. I did. So using that fancy instrument I keep talking about, the MRM, we used standards at the top to compare with our sample to see if we did have different sulfur bacteria. And we actually found a suite of them. So we found chlorobacterium, which is a green sulfur bacteria. We found openane, a purple one. And we found isorenitane, beta isorenitane, which are brown-green ones, green-brown, I should say. So we find the whole suite. The reason they're different colors is because they occupy different colors, different layers of the water column. And essentially you get purple, then you get green, and then you get brown. And they have different tolerances to oxygen. So the purples can be a little bit more tolerant of oxygen, but the brown ones, they wouldn't be happy at all and they'd die off. So that's the reason they have different layers, because it's that distance from the oxic layer of the lake. So we get a really prominent isorenitane peak. But I guess the great news about this is, yeah, we did have photic zone euxinia in the lake. Now, I'm only going to say this once. MTTCs stand for methyl trimethyl cyclochromans. Some previous work in the group identified them in the Go-Go Formation, actually. And essentially, it's a set of different compounds. You get alpha, gamma, and theta, I think. The ratio of the alpha compounds over the total of them, that tells you about the water column and how saline the upper water column was. So the fact that I have, this graph from a published paper in our group, this is 1, we notice this is 0, this is 0.8, so I was like, having to represent it on here wasn't nice, but it actually, even though it's down in that corner, it is actually quite a high value and tells us that we have a freshwater cap existing on the lake. So you'd have salty, sulfidic, no oxygen, and then a freshwater cap on the top. Yeah, similar to what we see in some of the Go-Go. So when we think about trying to think of a lake model of deposition in this fossiliferous basin, we have a small elliptical lake, and I think that really concentrates all the ions and nutrients coming off the land, and then you get that evaporation, concentrating it further. And the restrictive nature can also quite easily lead to stratification of the bottom waters. But there is geological evidence for a freshwater cap, but also no oxygen and sulfur, rich lower waters, and that lack of algal blooms as well. So I tried to kind of summarize it and how this could be good for fossilization in this way. But when we think about the wider project, what are important drivers for preservation? Probably ecological space for the organisms to actually live and thrive in the first place. If there are no organisms to preserve, that's it. You also want processes that inhibit rapid scavenging, and you want probably rapid burial if you can get away with it. And as I said, this is in a larger project to understand fossilization in my group. And my present work is actually destroying, stop saying the word destroy, geochemically analyzing the fossils themselves. So I've separated out different fish fossils, different components, and we're seeing different geochemistries associated with fossilized bits of fish. Skin, totally different to the vertebra and the thin bones, which are all different to the sedimentary matrix. So we're actually preserving the geochemical signature of potentially the fattier acids and the collagen of the skin compared to the other bits. And yeah, so my current work is more on the soft tissue preservation. We also have people in the group working on all these different fossils, ichthyosaurs from Germany, turtle bones, and people working on modern microbial maps, putting things in and seeing how they fossilize. So it's part of a big, wide project. But to really conclude with my work, how is this good for fossilization? Well, we have a suite of biomarkers that indicate you've got salinity stratification, you've got anoxia, and you've got photic cynic conditions. And that would really inhibit the degradation of the fossils through scavenger. So that's one point. You also are lacking these large algal blooms that are in all the other Green River basins, and you have a freshwater cap at the surface. So you have that ecological space for things to thrive that maybe the other lakes didn't. And the morphometric properties of the lake, that long, thin ellipse, narrow, would give you a narrow basin with a high shoreline index. So you get all those nutrients and ions inputted in and the water evaporated out. So I think with all of that together, you've kind of created this perfect storm for optimal conditions for fossilization. It's then getting buried rapidly through the carbonate precipitation, and you're just not getting that mixture of ingredients in the other Green River basins. Modern analogs is for shape, not for chemistry, I'd say. The shape could be the very warm place, Rift Lakes you see in East Africa. They're deeper and they're different. It's a different world, but morphometrically, that could be essentially, you can imagine how much input you're getting washed in. So I guess after that, I'd like to thank the Laureate Fellowship funding, and also Ansto, the John Delaney Centre, Tiger Curitn, and all the technical support and help from people, and Syro, who helped with the CT scanning. Lionel. And if anyone wants to read more, it got published a week or two ago in Organic Geochemistry. So thank you, and I'm happy to take any questions, and thank you for sitting through all that organic geochemistry.